Hi, everyone. Thanks so much for coming to my talk, and thank you to the organizers for inviting me here today. My name is MichelleHarwood. I'm a PhD candidate at the University of Toronto and at the Ontario Institute for Cancer Research. And I'm really excited to be here to talk to you about my research today, which is looking at recombination and population demographics and how they impact allele-specific expression in human populations. So to begin, I'd like to start my presentation by discussing forces of evolutionary change. So we have natural selection, genetic drift, mutation, and migration. Now, I'm specifically interested in natural selection because it's been really important for many areas in human genetics research, including pinpointing functionally important regions of our genome, the genetic adaptation to different environments around the world, and for the frequency and intensity of many human diseases. Now, natural selection is very well characterized in our DNA, specifically when looking at coding regions of our genome. However, we're only beginning to understand the role of selection on gene expression variability. And this categorizes a whole new avenue of our genome, including these regulatory regions. This is going to further influence these aspects of human genetics, specifically looking at how evolution is shaping our regulatory genome. So one way we can investigate natural selection on gene expression is through a process called AlleleSpecific Expression, or ASE. This is where we have the unequal expression of two alleles at a particular position, and this can be caused by a regulatory variant in cis. Now in this example here, we see where this is causing an overexpression of this damaging mutation, or we can see where the reverse where we have an underexpression of that damaging mutation. In fact, in 2018, Castal et al. Has showed us that selection acts to remove haplotype combinations that overexpress highly deleterious or damaging mutations. So this is showing that selection in combination with ASE has this protective effect. Question remains, though, of how this process works. So we know that selection is impacting ASE and we know the overall actions, but what in our genome is actually contributing to this? This is my research comes in where I'm investigating aspects of our genome that contribute to the efficiency of selection, specifically recombination and effective population size, to see how they influence the actions of ASE genome-wide. To do this, I'm using data from CanPath, which is Canada's population cohort. We have 1,000 individuals that have been genotyped and RNA sequenced from whole blood, and they have ancestry from Africa, Europe, and 3 subpopulations of Quebec, Quebec City, Montreal, and Saguenay. And all of these ancestral populations have different population sizes, which is what we'll be discussing later on in the presentation. I'm also using the Genotype Tissue Expression Project, or GTEx, to test for whether these results are tissue specific or seen across tissues. And I'm looking at whole blood, muscle, brain, and ovarian tissues. So to actually test for ASE, we've developed this pipeline, which starts with our genotyping array to identify heterozygous positions. Once those positions are identified, we can then turn to the RNA sequencing to look for those same positions and get the read count for each allele at those positions. We then correct for mapping bias. This is a known and very important bias in RNA sequencing data, specifically when looking at allele-specific expression, and we're using the WASP mapping bias correction here. We then can test for ASE using binomial tests, and this tests from deviations from 50% using the read counts. So if the read counts for each allele are equal or close to equal, this would be a 50-50 ratio, and this would show that it would be not significant. There's no ASE at this position. But if we have deviations where one read count is much higher than the other, this would test significant with binomial tests and will show evidence of significant ASE. We then correct for multiple testing because we're doing this for every individual and every SNP separately. So we have millions of tests. And then I use ancestral alignments to identify which allele is the ancestral allele and which is the derived allele. The ancestral allele is found in chimpanzee, rhesus marquis, and human genomes, and the derived allele is only found in humans, so it's the newer mutation. This is important for the directionality of ASE because moving forward, I'm going to refer to it as overexpression or underexpression of the derived allele. So to give you an idea of genome-wide ASE, I've tested approximately 3 million individual SNP pairs, and I've found that about 6% have significant ASE. And of that 6%, it's quite equal in terms of those that over and underexpress the derived allele. Now, moving forward in the presentation, I'm only going to be presenting those that are missense and synonymous SNPs, but they have very comparable estimates to the ones you see here on the screen. So this result shows the overall actions of ASE, and it supports the research by Castell et al. That I showed at the beginning of the presentation. On the x-axis, we're looking at CAT score. This measures the level of how damaging a mutation is. So a CAT score of 10 shows us the top 10% most deleterious mutations in the genome. CAT score of 20 is the top 1%, and a CAT score of 30 is the top 0.1%. And on the y-axis, I'm showing you the derived allele proportion, where anything above 0.5 is overexpressing the derived allele, and anything below is underexpressing the derived allele. And so what we see is this observed trend where we have this underexpression of highly deleterious mutations. And so now this is important because as I introduce aspects of selection, so recombination and effective population size, I want to see if this result changes at all. So starting off with recombination, recombination is really important because it reduces selective interference or Hill-Robertson interference at these linked sites. So in this example here, if we have a beneficial mutation and a damaging mutation on the same haplotype that are linked, selection acts to increase beneficial mutations, but would have to carry along that damaging mutation with it, or would act to decrease damaging mutations, but again, would have to carry along that beneficial mutation. So these are both not ideal scenarios. But if we have a recombination event, this can break apart that linkage and allow selection to act on those mutations independently. Therefore, regions of our genome with higher rates of recombination have more efficient selection. And since I'm interested to see how selection is influencing ASE, I can test to see if ASE is enriched in certain regions of our genome based on their recombination rate. And what I observe is that SNPs with significant ASE, regardless if they're overexpressed or underexpressing the derived allele, they're more likely to be observed in regions of high recombination and normal recombination compared to cold spots. And on the screen, I'm showing you here from CanPath or Canadian's cohort for whole blood, but it's actually observed across tissues. So we're looking at blood in GTEx, you can see muscle, brain, and ovarian tissue have very similar patterns in enrichment in these high recombination and normal recombination regions. So overall, this is suggesting that ASE is not randomly distributed along the genome, and that recombination rate seems to influence ASE variability. And it's likely because of the influence that recombination has on natural selection. So to briefly bring you back to this plot where we see this underexpression of highly deleterious mutations, but now I want to split this based on recombination. So we see regions of high recombination in red and low cold spots in black. And we can see that this observed model is still holding true in our high recombination regions where we're underexpressing highly deleterious mutations, but it's not holding true in our cold spots or our low recombination regions. We don't see the same effect. Interesting though, I also can split this based on expression. So this is again holding true only at sites with high expression, and this is looking at total expression. So only those with high expression is not observed in low and medium expression bins. So this is showing us that recombination in combination with expression, these both have high influences on the efficiency of natural selection, where higher regions of recombination and higher expression have more efficient selection. And this is also where we're seeing that it's more efficient at underexpressing deleterious mutations using this ASE. So next, since we've now identified this link between expression and ASE, we next wanted to see if tissue-specific ASE within an individual can be explained by these expression regions or the recombination regions. So to do this, I identified 79 individuals with measurements in blood, muscle, and brain tissue at 448 sites where ASE is observed in at least one tissue. So using this heat map, we're looking at columns are the individual SNP pairs and the rows are the different tissue types. And the colors are showing us if ASE is significant in each tissue and what direction they're observed in. And so we identify many, many positions of this highly variable ASE across tissues for the same individual at the same site. But looking at the bottom, we see the annotation of recombination, and it doesn't appear to be directly linked to recombination. However, when now I add expression into the mix as well, we see that sites with tissue-specific ASE, so those that have ASE only in one of those three tissues, that were highly expressed, have an increased odds of being observed in high recombination or normal recombination regions compared to cold spots. And this is not observed in medium and lowly expressed sites. So since higher recombination and higher expression have more efficient selection, this result is suggesting that sites under stronger selective pressures are more likely to exhibit tissue-specific ASE. The next aspect that I want to discuss today is the effective population size. So this brings the concept of genetic drift, where smaller populations such as Saguenay have more susceptible to effects of genetic drift and have less efficient selection compared to larger populations such as Africa. And so if we look at this odds ratio once again, where we see the enrichment of ASE in high recombination and normal recombination regions, but this time I stratified it based on population size, where the larger affected population of Africa have an increased enrichment in these high recombination normal regions compared to Europe, which has a smaller affected population size. And when I add in our populations of Quebec, we again see this reduction in enrichment based on these effective population size being reduced as well. And this is specifically observed in overexpression for our whole blood. But I've also replicated in muscle, we see the same trend between Africa and Europe, and we also see it for our derived underexpression bin as well. And so this is again supporting the idea that effective population size is also a really important aspect of selection, and this is likely also affecting our actions of ASE genome-wide. So today I showed you that these three aspects of efficiency of selection, recombination, total expression, and effective population size, seem to facilitate ASE evolution, specifically where sites with higher selection efficiency are using ASE to protect from highly deleterious mutations. And this is going to further influence these aspects of human genetics research that I touched on at the beginning. We can look at these in terms of genetic adaptations to different environments in these different populations, and also the penetrance of many human diseases. And with that, I would like to finish off by thanking my lab. Thank you so much to my supervisor, Philip Awadala, to my entire lab, but specifically Mary Julie, Fabian, Dave, Isabel, and Hillary. Thank you for listening. My contact information is there. I'd love to chat with you all more. And thanks again to the organizers for inviting me here today.